Hello everyone, and welcome to this Biology Mind A-Level tutorial on the structure of cell membranes. So just to recap, in the last lesson we learned about cell division and that there are many phases within the cell cycle. You now understand that cancer is uncontrollable cell division that arises from key mutations, and you understand the process of preparing a root-tip cell squash in order to observe the different stages of mitosis using a light microscope. So in today's session we have three main objectives. Number one, we want to go over an overview of the cell membrane structure, mind me with my pen. Number two, we want to look at the fluid mosaic model. And number three, major components of the cell membrane. So cell membranes are made of phospholipids. So this one structure here is a phospholipid. So this diagram is showing two phospholipids. Now, a phospholipid molecule has one hydrophilic polar phosphate head and two hydrophobic non-polar fatty acid tails. So just to make that a little bit clearer for you, this is our hydrophilic water-loving head, and these two, well, I'll do this in a different colour just to make it a little bit clearer, these two are our hydrophobic tails. Now, just in case you're struggling to get round what hydrophilic and hydrophobic mean, let's just make that a little bit easier by thinking about what the word actually means. So these words arise from Greek origins. So hydro has a Greek origin of water, and phobic, when you go back to its Greek meaning, is hate, so water hating. And then hydro again, water, philic, from the Greek of love, so water loving. And this is relevant, and you'll see why in a sec, because that all determines which way round our hydrophilic head and our hydrophobic tail is positioned within our membrane. Okay, so as I said, our head is our water-loving head, hence this nice little heart emoji here. And our tails are our hydrophobic tails, and they are water-hating. So now let's have a look at this in context. So this diagram is showing many phospholipids. Now remember, this structure here, so this one structure here, is our phospholipid. This is our hydrophilic head, and this is our hydrophobic tail. Now, it's important to remember that cells have an aqueous solution both outside and inside. So outside, the aqueous solution is called the extracellular fluid, and inside it's called the cytoplasm. Now, either way, these are aqueous solutions, which means that they are water containing. So now have a think back to what we said about our tails and our heads. So quite naturally, our hydrophilic heads, which are water-loving, are going to be facing the side where the water is, so i.e. where the cytoplasm and the extracellular fluid are. Whereas our hydrophobic tails are water-hating, so they're going to hide away from our extracellular fluid and cytoplasm. So they're going to be tucked inside of this layer here. So you can see that our hydrophobic tails are tucked inside nicely here, away from our extracellular fluid and our cytoplasm. Now, the phospholipids form a bilayer, so this is a really important word that you need to commit to memory. It's a phospholipid bilayer, that's what we call our cell membrane. And as I said, hydrophobic tails on the inside, hydrophobic tails are facing out on the outside. And remember I said that this is the cytoplasm here, and this is the extracellular fluid. So hydrophobic heads of the phospholipid face the extracellular fluid and the cytoplasm on the inside. This is our phospholipid bilayer, this whole structure. These are our hydrophobic tails, these are our hydrophobic heads. And as I said, the hydrophobic tails are folded into the membrane and protected from water. So now this brings us quite nicely onto what our fluid mosaic model means. So the fluid mosaic model describes cell membrane structure. So when we say it's a fluid model, we mean that the membrane itself contains these phospholipids that are constantly moving around the cell or the organelle. And by mosaic, we mean that proteins and other molecules, such as cholesterol, are embedded within the cell membrane, as we can see here. So we'll take a closer look in a sec. So these are our fluid proteins. They're present all around the circumference of the cell, and they can move sideways across the bilayer. These are our stationary proteins, and they are anchored to a particular position, and this is often seen with many types of receptor and transport proteins. Okay, so that's a summary of our proteins and why they are fluid. So these are our fluid proteins. These are our stationary proteins. So we can see that in terms of the fluidity of proteins, we have some that are fluid and move about, and some that are stationary. Okay, so in terms of the other major components of our cell membrane, so remember we've already talked about our phospholipid bilayer. We've already talked about some of our proteins. We also have our glycoprotein, which is a protein with a carbohydrate attached. We have our glycolipid, which is our lipid with our carbohydrate attached. So easy ways of remembering this is glyco means carbohydrate. Glyco means carbohydrate. So here we're talking about a protein with a carbohydrate attached, and here we're talking about a lipid with a carbohydrate attached. So that's quite nice and self-explanatory there. And then we have these channel proteins, as discussed earlier. We have cholesterol scattered throughout the phospholipid bilayer as well. So cholesterol helps reduce the fluidity of the phospholipids because we need our membranes to have quite a nice rigid structure in order to protect our cells and also to provide a definitive structure to our cells. So this is how our cholesterol embeds within our phospholipid layer. So just thinking a bit more about cholesterol. Cholesterol itself is small, and as seen on the previous diagram on the last slide, it's usually found squeezed in between the tails of phospholipids in the phospholipid bilayer. Now, in terms of its function, it adds to the hydrophobic barrier. So cholesterol is hydrophobic. Adding to this hydrophobic barrier, it restricts movement of phospholipids, making the membrane more rigid and complete. So it adds to that barrier function of a membrane. It also increases thermal stability, so cholesterol prevents the membrane becoming too fluid when temperature increases, or too rigid when temperature decreases. Now, channel proteins extend right through the membrane, forming a passage for ions, which will meet at a different point in the course. Carrier proteins move freely around the membrane and actively transport molecules. Receptor proteins act as receptors for cell signalling, and carbohydrate chains can attach to membrane proteins, making glycoproteins. Carbohydrate chains can also attach to membrane lipids, making glycolipids. So this is a nice summary of all the various functions of our proteins in our cell membranes. So let's take a closer look at this now. So remember we said our channel proteins extend right through our membrane, forming a passage for ions. Our carrier proteins move freely around the membrane and actively transport molecules. And our receptor proteins act as receptors. So now we can think of function of proteins within our cell membrane. We know that number one, the function is ion transport, because remember we saw that we have these channel proteins that allow for ions to move from the outside to the inside of the cell and vice versa. And number two, large molecules. So carrier proteins transport large molecules by binding to the molecule, changing shape and releasing the molecule on the other side. Now let's think about our carbohydrates. So we have our glycoproteins and we have our glycolipids. Now, in terms of functioning, number one, they provide the function of being a binding site. So they can detect signaling molecules and can be binding sites for hormones, antibodies, and drugs. So, for example, they act as antigens for antibodies to bind. In terms of our second function, it would be hydrogen bonding. So glycoproteins and glycolipids form hydrogen bonds with water molecules, which you'll meet in chemistry, strengthening the membrane. And then number 3 is cell adhesion. So glycoproteins can help 2 cells join together to form a tissue. And as we know, many cells form tissues, and many tissues form organs, and many organs form systems. Alright, so that's our lesson complete for today. Well done for following through till the end, and I'll see you for our next session.